chapter is about cell division, and basically all the cells that are in our body, whether they are the cells from the immune system, from the transport, from muscles, from the brain, from hair, from skin, from any body part, right? They are basically called the autosomal cells, right?
Right.
The cells of the body. So these autosomal cells, they do not have like life, a very long lifetime, right? Lifespan. They have a very short lifespan, which means that after a certain time, these cells will have to be replaced by new cells. Fine. So the
Fine.
replacement of this new cell occurs by a process which is called cell division. So this cell division basically means the older cell is going to divide to produce more new cells, right? Okay.
Right. Yeah.
So, for example, the red blood cells, they have a lifespan of about 120 days. So after 120 days, the red blood cell is going to divide and it is going to form new cells. Similarly, the white blood cells, they usually have a very short lifespan of around few hours. So after every few hours, they are replaced by new ones. Fine. So
Fine.
the process by which these autosomal or the body cells divide, the process of their cell division, this is called mitosis. So what is basically mitosis? So every cell that is present in a body has some DNA and has some chromosomes, right?
Yes.
And so in every living organism, whether it's plant or whether it's animal, whether it's human, it has some nucleus and some chromosomes in it, and it has a fixed number of chromosomes. That means that it has a fixed number of DNA content in every cell. So all these cells are going to have the similar content, similar genetic content, right? For
Right.
example, in case of humans, there are 23 pairs of chromosomes, right?
Mm-hmm.
So 46 chromosomes in total. 23 pairs means 46 chromosomes, which is actually the genetic material. So in every cell, there is going to be 46 chromosomes in the nucleus, right? And
Fine.
every time this cell divides, it needs to copy this 46 number of chromosomes to the upcoming cells. It needs to copy all the genetic material to the next copy. Fine. So this is how it happens. This is a parent cell. That means the cell that has to divide. Parent cell means the cell that has to divide. Fine. So this parent cell
Fine.
is going to duplicate its DNA, which means it's going to double the amount of its DNA just before the division. So if it has, for
Mm-hmm.
example, this 4 chromosomes, it is going to make them 8. Fine. Now, what
Fine.
is going to happen now? These 8 chromosomes are now going to get dispersed in this nucleus, and like they are going to equally divide into 2 cells. So this parent cell has actually duplicated its DNA content first and divided into 2 new cells now, which are called daughter cells. Fine.
Fine.
So now these
Yeah.
2 daughter cells will have half amount of this part of the cell, this part of DNA, right? So half amount
Mm-hmm.
means this is going to have 4 chromosomes and this is going to have another 4 chromosomes. So now the number of chromosomes in daughter cells is equal to the number of chromosomes in the parent cell. You see,
Yes.
it became 8 from 4. The 4 turned into 8, and again that 8 split into 2 daughter cells with 4 chromosomes. So basically, mitosis means the division of cells, but it does not involve reducing the number of chromosomes. The division of cells will not lead to reduction of the number of chromosomes. The number of chromosomes will remain same. Fine. The division of cells without changing the number of chromosomes. Fine.
Fine.
Every time, let's say a red blood cell divides, it will divide in such a way that the daughter cells are also going to have the same genetic content. Similarly, for the muscle cell, nerve cell, white blood cells, and everything. Fine.
Fine.
But the cells which are related to heredity or which are related to reproduction, fine, which are related to producing a new organism, they are these cells, sperm and egg cell. Fine.
Mm-hmm.
These cells, they are going to produce a new organism. Maybe it's a plant or maybe it's an animal by fusion of 2 different cells, right?
Fine. Yeah.
So if this cell has 46 chromosomes, let's in case of humans, if this has 46 chromosomes, and this also has 46 chromosomes. The fusion of these two will lead to double number of chromosomes, right?
Yeah.
So technically, what should happen here, that these two cells should have half the number of chromosomes in each, okay? So this
Okay.
should have 23 chromosomes and this should have 23 chromosomes, right? And then when
Right.
they fuse together, the new human body, which is going to produce from these two cells, should have 46 chromosomes, right?
Right.
These sperm and egg cells, or the sex cells, they actually have half the number of chromosomes from the number of cells from where they are producing. So whatever cells are producing the sperm cell, or whatever cells those are producing the egg cells, will be having 46 chromosomes. But when they split up or when they divide, they will give only 23 chromosomes to this one and 23 to this one. Fine.
Mhm.
Now,
Fine.
this process of reducing the number of chromosomes after cell division is called meiosis, right? So now,
Okay. Yeah.
here, this parent cell is actually the cell which is producing, for example, this egg cell. So this is the
Mhm.
parent cell, the cell that will divide into this egg cell. Now, what will happen? It will double its content first, right?
Right.
And then it will split into two. So now this stage is called meiosis 1, the first stage of meiosis. So in the first stage of meiosis, you'll get the similar number of chromosomes, fine, and you'll have
Fine.
two different cells. But there is an additional step. In this step, these two cells are going to then again divide. Now this is 4 and this will contain 2. So 2 will go here, 2 will go here. So you see that
Mhm.
from the parent cell, the number of chromosomes in the final daughter cell that is going to be produced is actually reduced. This
Mhm.
parent cell had 4 chromosomes and the daughter cell will be having 2 chromosomes,
Two.
right? So
Yeah.
this parent cell will be producing 4 different types of daughter cells. Why 4 different types of daughter cells here? In
Mhm.
case of mitosis, these two are identical. These two daughter cells are similar to each other. But in case of this meiosis, these 4 daughter cells are very different from each other. How? I'll explain it in a while, that during this meiosis 1 stage, there is actually a recombination of chromosomes so that the genetic variation is introduced. So every time an egg cell is produced, it is not identical to the mother's cell, right?
Mhm.
So that's why we have genetic variations. The mother is not exactly similar to the daughter, or the son is not exactly similar to the father, right? You have
Right.
genetic variations. So those genetic variations are actually arising from this phase of meiosis. Here, there are
Mhm.
some combinations. There are some steps which involve shuffling of the genes, shuffling of the chromosomes between cells, and then this produces the egg cell. Now, one genetic variation comes from this phase of meiosis, and another variation comes from combining of these two different types of cells together. So the
Mhm.
genetic variation is enhanced in two different steps. First step is the genetic variation caused by the recombination here, and the second step is the fusion of two different types of cells. So this 23 number of chromosomes is going to have entirely different set of genes.
Mhm.
It is
Yeah.
coming from another organism which has completely different set of genes, and this egg cell is going to have completely different set of chromosomes. So when they fuse together, they have like a different combination of genes, fine. While in case of
Fine.
mitosis, every time both the cells are going to have same chromosomes and same number of chromosomes. So there is no variation in the genes and there is no variation in the number of chromosomes, right?
Right.
So these are the two major differences in mitosis and meiosis. Now, how the cell divides? This is called the cell cycle, and there are different phases of this cell cycle. Fine. So
Fine.
this interphase is that phase when the cell is actually not dividing, or when it is just growing and just moving around and performing its function. But when the cell decides to divide, right?
Mhm.
What happens is that it starts entering this G1 phase. Fine. So this G1
Fine.
phase is actually it's thinks about dividing, and it starts duplicating its chromosomes and its cellular contents, let's say. It starts duplicating its organelles, and all the ribosomes, all the plasma, or, sorry, all the cytoplasm and other organelles. Everything that is inside the cell is going to duplicate in this G1 phase. The moment the cell starts thinking to divide, it starts duplicating its content, right? It is the first step of preparation for division. Okay.
Yeah.
Now, after this G1 phase, what is going to happen is that in this phase, all the proteins and the organelles will double. Now, in the S phase, the chromosomes are going to be duplicated. So, all the 46 set of chromosomes is going to be duplicated. Fine.
Fine.
So, now the whole genetic content has been duplicated. Now what will happen is that the cell is going to the final stage, which is called the double checking stage, where the cell needs to check that everything is perfectly okay for division, and you do not need to repair anything before division, because if error happens at this stage, it is not going to be reversed. The cell with error is not going to perform its function. Fine. So to avoid that, the cell is going to double check at this stage that everything is perfectly fine for division, and then it enters this division phase. Now this phase is the division phase. This one was only the preparatory phase. Okay.
Okay. Yeah.
So this one is preparatory phase for division, and this is actually the division phase, whether it is mitosis or whether it is meiosis. Okay.
Okay.
So in this phase, the cell is going to divide all its contents, all the duplicated chromosomes and everything, the cytoplasm, right? Not the cytoplasm, just the nuclear content. The cell is going to divide its nuclear content, its chromosomes, etc., right? Now, after duplicating its chromosomes, the cell is going to divide its cytoplasm. The cell is actually going to split between the two. Fine. So this was the cell. Then after meiosis, it has transferred its nuclear content to two different cells. So the cell has actually pinched inside. I'll show you the diagrams in a while. And it has actually converted into the two daughter cells. So this is what is happening in the cell cycle. Now you see a G0 phase here, or a G0 cycle here. So what happens in some cases is that this cycle is happening in all the body cells. Every time a cell is produced, it starts duplicating its content, double checks, divides, and then these two cells go through this process again. Fine. Every cell is going to go through this process, right? But there are some cells that after one cycle of division, they quit the cycle and go outwards, which means that once formed, these cells are not going to divide again. Right?
Yeah.
These cells are not going to divide again. The cells that are divided are actually involved in multiple functions of the body, which are related to growth, which are related to your, you can say, transport. Fine. So, however, some cells, there are some cells in the body that do not need to divide further. For example, the cells of your eyes. Fine. So your eyes, they have a typical size from your birth till death. So that once their cells are divided, they remain the same throughout your life, right? Similarly, there are neurons in your brain that actually enter the G0 phase. So there might be other examples as well where the cell goes to the arrest stage so that it does not divide. Or maybe there are some cells which go to this phase for a certain period of time. Maybe for some years they do not divide, and after that they start dividing, right?
Yeah.
So this G0 phase is actually the cell cycle arrest, which means that the cell is not going to divide, it is not going to die, it is going to, or if it dies, it is not going to be replaced by new cells. Fine. So it remains as such. Fine.
Fine.
So this is the cell cycle phase. Any question up till now?
No.
Okay. So this is mitosis now here, and this is like as I told you, it is happening in all our body cells, and this is just an outline of how mitosis happens. So this is interphase. Basically, the chromosome has duplicated, and the centrosome is the structure that is involved in division of this nuclear content. Fine. So once the DNA is doubled, the next phase that comes, the first phase of mitosis, this is called prophase. Fine. It has this
Prophase, yeah.
typical name, prophase. In this prophase, what is happening is here is that all the duplicated content, it is going to pair up. Fine. It is going to
Mm-hmm.
form pairs. Now, once it forms pairs, it will decide that this content, the content of genes on this chromosome and the content of genes on this chromosome is equally going to divide between the two cells. So that is why they pair up. Fine.
Mm-hmm.
Now, in the next phase, when they enter, this centrosome is going to move to the opposite poles and it starts creating microtubules or smaller tubules or fibers that spread across the cell. Fine. When it
Mm-hmm.
spreads across the cell, these duplicated chromosomes which were paired up actually line up on this plate, on the center of the cell. This center of the cell is called the plate, central plate. Fine. The metaphase
Yeah.
plate or the equator, metaphase plate or the equator. So this is going to line up here because these fibers are going to attach with these chromosomes, right? Now, once
Mm-hmm.
they attach with these chromosomes, they are going opposite poles of the centrosomes. They are going to pull both these centromeres, chromosomes towards themselves, right? So they are going to
Right.
put a pressure, pulling pressure on these chromosomes, and because of this pulling of the chromosomes, the chromosome is going to split from the center. So half of this chromosome goes to one side and other half goes to the other side. So it has the same genes on it. So half genes went there, half genes went there, right?
Mm-hmm.
This is called anaphase. Sister chromatids, the two parts of the same chromosomes, those are split equally between the two parts of the dividing cell, right? Now, and
Right.
in the final cell, this is the telophase. Now, in telophase, actually, the sister chromatids which are pulled apart are actually, you can say, actually assembled in the nucleus in a new nucleus. This nucleus was like dissolved in the prophase stage. You can see here now there is no purple color. This purple color is representing the nucleus. So, first of all, the nucleus dissolves, and all these things
Mm-hmm.
are happening in the cytoplasm. Now this again forms a new nucleus in the telophase phase, and the cytokinesis means the cytoplasm is going to divide. So once the cytoplasm finishes dividing, the two identical daughter cells are formed. So this is a diagrammatic representation of how it is happening. You see here in the prophase stage, fine, the chromosomes are going to pair up after duplication. So
Mm-hmm.
these were single chromosomes. They doubled and then they paired up. Fine. These are called homologous chromosomes. I'll explain it in a while. Fine. Now these are two centrosomes. They moved to the opposite poles, started creating these spindle fibers, and the chromosomes have started gathering at the equator. Now these strings have attached to this centrosomal part of the chromosome. Okay, or
Right. Yeah.
centromere. If I draw a centromere here, it looks something like this. So the chromosome actually looks something like this. This is like fine. So this is something like this. So this central part is called centromere. Fine. This one.
Fine.
This central part is the part where the spindle fibers are going to attach. Right?
Yeah.
These
Yeah.
fibers are going to attach at this part, and when these pull the centrochromosome towards itself, both these parts are going to try pulling them it towards itself. But because this pressure on both sides is equal, this chromosome cannot go to any one side. What will happen here is that it is going to split from the center. So this half part is going to go towards this side, and the other half is going to go towards this side. Fine. So this is
Fine.
what is happening in the anaphase stage. After anaphase stage, the chromosomes which were pulled on both sides are going to develop a new nuclear envelope. Fine.
Mm-hmm.
So once this nuclear envelope develops, the cell is going to start pinching inwards, and it is going to split, which is called cytokinesis. Now this new cell is again formed. Fine.
Fine.
So this is called a diploid cell. Diploid cells means two daughter cells are formed that has same number of chromosomes, right?
Right.
Did you understand all this point?
Yes.
Was it like easily understandable, or is there any confusion up
Yeah.
till now?
Oh no, no problem so far.
Okay, so this is about mitosis, and every body cell is actually dividing through this way. The whole genetic material in the body cells are dividing in this way. Fine. So,
Yeah.
but you see there, I said that the cytoplasm is just going to pinch inwards and divide. But we know that in case of animal cell, you have only plasma membrane, and it can actually pinch inward and split into two. So this ring that is formed in the center when the plasma membrane is dividing the cell into two is called a contractile ring. Fine.
Yeah. Mm-hmm.
So this contractile ring is actually made up of different types of proteins which align here, and they are going to signal the plasma membrane to invaginate here. So once it starts invaginating here, the cytoplasm is going to divide and the cells are going to split. But in
Mm-hmm.
plant cells, we know that we have a very rigid cell wall, right?
Yeah.
It is
Right.
very difficult to divide the cell wall by just pinching inwards. So what is going to happen here is that once all the chromosomes have gone to the opposite side, at that time, all the Golgi vesicles in the cell are going to align again at the equator. Fine. Now,
Fine.
once these start aligning at the equator, these Golgi apparatus are going to help in dividing of the cell wall. So now this cytoplasm is dividing along with formation of a new cell wall in between this cytoplasm through with the help of these Golgi vesicles. So now
Mm-hmm.
both these cytoplasms have developed a new cell wall in between because of these Golgi vesicles. So this is how the plant cell divides through mitosis. It is very important to divide the cell wall as well. Okay, and the cell wall
Okay.
cannot divide through just pinching inwards.
Mm-hmm.
Now, this is second type of cell division, which is called meiosis, and it is happening in the cells of reproduction. So I told you there are two different phases of meiosis, which is called meiosis 1 and meiosis 2. The names are same, just a few steps are different, rest of the things are same in both. So similar to the prophase in mitosis, the nuclear envelope is going to disperse here, the nucleus is going to duplicate here, and the chromosomes are going to pair up.
Fine.
Fine. And then in the metaphase stage, they are going to align at the equator, right? Now,
Right.
something different which is happening here is that when these are not homologous chromosomes. Now, homologous chromosomes mean two same chromosomes. Fine. I've drawn it here, right? So these are homologous chromosomes, right? Which means
Yeah.
they have same size, same structures. Now, what happens here is that when they are aligned at the equator, these chromosomes are going to exchange their genetic material by simply crossing over here, right? Any two
Yeah.
chromatids, these are called two sister chromatids, and these are called two non-sister chromatids, right? So these are called two non-sister chromatids, and this blue one is called sister chromatid. Chromatids on the same chromosome are sister chromatids, and chromatids on the different chromosomes are called non-sister chromatids. So these two non-sister chromatids are going to exchange their genetic material, right? So now this one has its part and this one has its part, right? So this is called recombinant chromosomes or recombinant chromatids. So this
Mm.
is leading to variation. This is leading to the genetic variation here. Fine.
Right.
Now, these
Fine.
two chromatids are the recombinant ones, while the other two are similar to the parent chromatid. They haven't exchanged their genetic material. So, this is called the homologous chromosomes. So, now here, after aligning of the homologous chromosomes, the crossing over will occur. Fine, and the crossing over will lead to exchange of genetic materials at this metaphase 1 stage, right?
Right.
Now, at this metaphase 1 stage, after they have exchanged the material, again similar to the anaphase in mitosis, this anaphase 1 in meiosis, the whole chromosome is going to be pulled on the opposite sides now, not the chromatids only, because the pair of chromosomes are aligning in meiosis 1. In case of mitosis, there was only single chromosome that was aligning at one spindle. Now, at one spindle, there are two chromosomes that are aligning. Fine.
Fine.
So
Yeah.
now what is happening here? It is going to pull one chromosome at its side and other at this side. Fine. So now the homologous chromosomes are being separated from the equator in the anaphase one, and this new nuclear envelope that is developing across the two different sets of chromosomes is actually giving rise to two different daughter cells with genetic variability, right?
Right.
Now, this cell is going to have half of the material similar to parent cell and half of the material which is recombinant, right?
Mm-hmm.
Then it splits into two, fine. And now these two cells are going to enter the mitosis, the meiosis 2 stage, fine.
Right.
So what is
Mm-hmm.
going to happen here? Again, it is similar to mitosis. Similar to mitosis. This one is similar to mitosis. The difference comes here. Fine.
Mm-hmm.
Now these two cells are going to form another spindle. Now it has like this single chromosome in the form of two sister chromatids. Now in this, on the equator, one single chromosome or recombinant chromosome is going to pair up. Fine. Now, in every chromosome, one chromatid is recombinant and other is parental. Okay?
Yeah.
Did you understand this? Like this chromatid is recombinant and this one is parental. So if it lines up on the
And,
equator, one chromatid is going to be recombinant and another is going to be non-recombinant. Okay? So this is
yeah.
what is happening here. On the whole equator of both cells, half of the chromatids are parental and half of the chromatids are recombinant. Now, in anaphase 2, when these two are pulled together, now you see this side is non-recombinant and this side is recombinant. Fine.
Fine.
Similar is happening here. Half of half of the side is having recombinant set of chromosome and half is having non-recombinant chromosomes. Now this cell and this cell are going to split into four different cells. Fine. When these four cells are produced, you can see that you had eight sister chromatids here, fine, or eight chromosomes here, and in the cells produced, you have four chromosomes. So the number is reduced. The number is reduced to half. Fine. And you also got 50% genetic variation in the new daughter cells produced. Did you? Because half of this and half of this was actually like parental cell and rest of the halves were equal to the new recombinant DNA. So
Yeah.
not even the set number of chromosomes reduced, you also got genetic variability. Okay?
Mm-hmm. Yeah.
So why are these exchanging these sister chromatids? Because if you have a chromosomes with two different sister chromatids and both of these, let's say, sister chromosomes, they can have different types or different versions of the gene. Let's say there's a gene for tallness. So there's a gene for tallness that also controls shortness, right? There's a gene for height. So one is controlling tallness and other is controlling shortness. So these are the two different versions of the same gene, the gene for height. Fine.
Fine.
So these alleles are going to lead to genetic variation. Let's say this blue one is having genes for tallness and this red one is having genes for shortness. Okay. So
Okay.
now they are going to exchange materials and they're going to decide which daughter cell from this one is going to fuse with the sperm or maybe the egg to finally decide whether the baby is going to be tall or short. Okay.
Okay.
The combination of this gene is actually producing all the variation in heights, weights, skin colors, intelligence, and everything. Okay. All the
Mm-hmm.
variation that you see in the world is because of this step in meiosis. Okay. The crossing.
Okay. Yeah.
Okay. So any question up
Right.
till
No.
now? Okay. So once these cells they have divided the body cells and the reproductive cells or the sex cells are going to help in production or and growth of the organism. So basically, there are different levels. This chapter has nothing important, just to understand different levels of organization. So once the cells divide, they're going to divide and increase in number, and they're going to produce a tissue. So this is a plant tissue and this is an animal tissue. So these cells are going to produce tissues, and different types of tissues will combine together to form an organ. Let's say these are the cells of heart. Fine.
Mm-hmm.
These cells of heart are going to produce the smooth muscle tissue. Fine. Or cardiovascular tissue. This tissue for heart muscles. Fine. So this is a tissue, and this tissue along with other cells, maybe the vessels, maybe the blood cells and other cells, are going to combine and then form an organ, right?
Right.
Now here the stromal cells, they have combined to form the leaflet. Fine. Now these different organs, they're going to combine to produce an organ system. Organ system means different organs are working in collaboration to produce a function, to perform a function, right? So this leaf
Right.
is working with a stem and another leaf to perform photosynthesis, maybe. Fine.
Mm-hmm.
And this heart is going to work with blood vessels, let's say, for transport. So one organ is working with another organ, let's say blood vessels, to perform a combined function. Fine.
Fine.
Now, these different organ systems are going to combine to produce an organism. Let's say along with this heart, you have lungs, you have skin, you have kidneys, you have brain, you have other organs, right? So all these are different organ systems. So these organ systems are going to combine into a whole new organism. Similarly, this leaflet structure is going to combine with root, stem, and it is going to produce a whole new plant. So these are the different levels of organization in any living organism. The cells, they combine to form tissues, and then the tissues, they are combining to form an organ, and then different organs work together to produce an organ system. For example, your digestive tract, it involves different organs, so it is actually an organ system. The digestive tract, it involves your mouth cavity, it involves your stomach, it involves your pharynx, it involves your trachea, it involves your intestines. There are multiple organs, right? It is a whole organ system. Okay. And
Right.
this organ system is working with other organ systems like the heart organ system, lungs organ system, brain organ system, to produce the whole organism, or to complete functions of the whole organism, right?
Mm-hmm.
Moving ahead, this was a chapter about stem cells and differentiation. There are different terms. So stem cell is basically any undifferentiated cell in the body, any cell in the body that has the capability to produce any other cell or any other tissue. Okay, so this is called stem
Mm-hmm.
cell. So when the life starts, you actually have some cells that do not have any specific name. They can form any other cell. Let's say when the sperm and egg divide, fuse together, they form a zygote, and then that zygote is going to produce the whole organism. So how it is
Mm-hmm.
going to produce the whole organism? By producing different tissues, by producing different cells. So it has the capability to turn into any cell, right?
Mm-hmm.
The stem cell is undifferentiated, means it does not has a specific fate to decide which it is going to become finally. It can turn into an any cell, right? So this is called the
Right.
stem cells. So these tissue cells, these are differentiated cells. Now this muscle cell cannot be a heart cell. This muscle cell cannot be a cell for blood. This muscle cell cannot be a cell for brain, right? This immune cell cannot be a cell for bones. This immune cell cannot be a blood cell, right? So basically, differentiated cells are those cells that perform a specific function only. However, stem cells are those cells that can perform functions of any cell, or that can be transformed into any other cell. Okay, did you get it?
Yeah.
Okay, so based on this stem cell, we have three different terms, which is called totipotent, pluripotent, and multipotent. So a totipotent cell or a totipotent stem cell is any cell that can differentiate into any other cell type. So basically, this one is a totipotent cell. It can differentiate into any other type, right?
Right.
Now, pluripotent arises from this stem cell. Pluripotent is a cell that has arisen from the totipotent cell. So it can
Mm-hmm.
differentiate into cells from any of the three germ layers. So basically, you have ectoderm, you have endoderm, you have mesoderm. So it can produce specific tissues only. Okay. This cell can produce specific tissues only. So we can say that, let's say this stem cell, it can produce a variety of tissues. So this is called the pluripotent cell, which has a multiple capacity of forming multiple tissues. Fine.
Fine.
Cells from multiple tissues. So this stem cell is actually pluripotent because it can form muscle, it can form fat cell, it can form bone cell, it can form blood cell as well. So a pluripotent is a subtype of totipotent cell, right?
Fine. Yeah.
Multipotent again now is a cell of a tissue, of a specific tissue that can differentiate into a limited range of cell types. For example, you had these muscle cells and there are some multipotent cells. They can only produce certain types of muscle cells only. Fine. They will not be
Yeah.
able to produce the bone cells or blood cell or nervous cell or anything else. They are going to produce certain types of muscle cells only. They can differentiate, but they will always form these muscle cells whenever they differentiate. Okay. So these are called multipotent stem cells. Fine.
Fine.
For example, the zygote, it can form a variety of other cells. So the zygote is basically a totipotent cell. And now from those different cells, let's say on the zygote, then turned into blastula and everything. And then those cells can produce different organs or different tissues of the body. Now those tissues can produce a certain type of other cells which are related to those cell types only. If I explain it like this, I think it will be more clear. So you see here, the zygote is totipotent. It can form any type of the cell underneath it, this pyramid. Fine.
Mm-hmm.
It can form any cell. Fine. Now this embryo, which is formed from the zygote, is actually pluripotent. Now embryo, it can form different tissue systems, different stem cell, different tissue systems, or even differentiated cells as well. So from this embryo, it can be a girl, it can be a boy, it can also form eyes, it can also form ears and everything. Fine. So this is basically pluripotent cell. Fine. It can form cells of different types of tissues. Okay. Now, multipotent stem cells coming downwards towards the pyramid. Let's say you have tissue stem cells. So there are specific stem cells for heart. There are specific stem cells for stem cells mean undifferentiated cells. So whenever you need those, for example, you have a cut on your skin, or maybe you might have seen lizard producing its tail again and again. Fine, which is called regeneration actually. So there are specific cells in the body. So whenever you need them, they rush towards that site and transform it or differentiate into those specific cell types. So if you have a cut on your skin and you need new skin cells, or maybe you have a burn on your skin and you need new skin cells. So those new skin cells are going to be produced from these stem cells actually, the undifferentiated ones. They are going to turn into skin cells, right? So if these stem cells are able to produce the cells of skin only, they are called multipotent cells, right?
Right.
Fine. But if they
Fine.
can, if these stem cells can produce skin cells as well as blood cells as well as heart cells, so they will be called the pluripotent cells, right?
Right.
Now, the unipotent cell is a unipotent means only one potency. So, if the cell is finally differentiated into any one type, for example, it has differentiated into blood cell, it has differentiated in muscle cell. Now, whenever it will divide, it will always form a blood cell only. It has only one potency of forming a one type of cell. So, that is called the unipotent cell. Okay.
Fine. Yeah.
I hope this is
Mm-hmm.
clear and does not cause any confusion.
No, not yet. No.
Okay. So now talking about different sources of these stem cells in animals and plants. So basically, the first source of these stem cells, or you can say undifferentiated cells, is when the egg and sperm combine to form a blastocyte or a fetus, right? In case of humans, if we
Right.
talk about it. Fine. So
Mm-hmm.
this fetus
Right.
is actually undifferentiated at this stage. It can turn into a human as well as it can turn into any other type. Fine.
Fine.
Okay. So now this has cord tissue. Fine. It is a rich source of all the stem cells. Now these stem cells will decide what organs to form, what tissues to form. So basically, the first source of the stem cells in humans is actually the embryonic cells or the infant stem cells, right?
Right.
If we need any stem cells from research, we can this type of stem cells are the most reliable, as they are like the most totipotent cells, means they can turn into any tissue type. Okay. Now,
Okay.
if a human grows up and becomes an adult, we still have stem cells in our body. As I told you, that we continuously need a replacement of different types of cells in body. For example, I told you red blood cells are replaced after every 120 days. The neutrophils and other cell types are replaced after every a few hours. You need stem cells to be transformed into the final differentiated cells. So the source of stem cells in adults is actually adipose, the fat tissue, the fat cells in your body, your bone marrow, and your tooth pulp as well. So all these have different stem cells, so they can rush towards the site where they are needed. Fine?
Right.
Do you get it? It's
Yeah.
just about reading that reading that paragraph about sources of animal cells, and I just gave an example that the
Mhm.
infants and the adults, that some types of adults stem cells are also involved in providing the store or the source of the stem cells in adult. So this adipose, this bone marrow, and this tooth pulp is actually the store or the source of the adult stem cells, right?
Right.
In case of plants, there's a tissue which is called the meristem tissue. It is actually present or involved in the growth of the plant. So it is basically at the tip of the shoots or at the tip of the roots. Fine, because roots are the ones that grow, or either shoots are the ones that grow. So this is called the meristematic tissue. This meristematic tissue is actually a rich source of stem cells. You see here, this is the tip of the root or maybe the shoot. So this tip is going to provide undifferentiated cells for further growth. Okay.
I see. Yeah. Yeah.
Okay. So from this
Mhm.
tip is going to produce a root or maybe a shoot or maybe a new leaf. So this tip has totipotent cells that are going to be transformed into either leaf, either root, either stem, whatever the plant needs. Okay. So this is
Mhm.
a source of stem cells in plants.